Okay, so my name is Guillaume Larocque. I'm a research professional at the Quebec Center for Biodiversity Science. Etienne approached me, I guess it was something like a year ago, and he said, Guillaume, we have a lot of data now in CABO, and the data is available in many files, and now the processes for accessing the data is through R scripts and other software, and we have to try and write a series of scripts to bring all those files together, produce the spectra and everything. And it would be great to facilitate data exploration and sharing by developing a portal to a web portal to query and visualize the spectral plant and trait data for the current CABO members, but also potentially for outside users when we're ready to start sharing the data. And that seemed like a great, interesting challenge. So I joined on board in beginning of April. So this has been my quarantine project over the last few months. I have to say I was extremely pleased to start working on this project, and I was extremely pleased when Etienne started describing all the data to me and all the data structure, because this is by far the project I've worked on where the data is best organized. And part of the reason of that for this nice organization is that all the field and lab data is collected in the Fulcrum mobile data collection system, which makes all the data very standard and very well structured. So when I started putting together a framework or a structure to develop this portal, so we had all these different forms available in Fulcrum, and then we quickly realized that it was possible to extract all the Fulcrum data to export into a relational database, such as Postgres and PostGIS. And then it was relatively straightforward to transfer all the data collected through the forms to the CABO web portal, to the CABO server. And then I wrote a series of scripts that easily took all the spectral data that was available in a series of text files, and I was able to import this really easily into the database as well. So then I built what's called an application programming interface to interact with the data through the web and through the interacting with the data potentially through R or other software like this, and specifically to allow web queries that could feed into the portal. So then the API was developed in Node.js and SQLize, and then I started developing a web portal in Vue Js, which is a web development framework that's pretty popular these days. So without further ado, I'm just going to give you a quick demo of the portal. So right now it's password protected. If you want to get access, you can ask Carol or you can ask Etienne or me. I think the password is also available on Slack, on the Slack channel. So the main way by which you interact with the portal is through the search bar on top. I tried to do a design that was as easy as possible. So for example, we can start searching by species, which is the main way to search the portal. And we can say, okay, we're going to do Acer and Ngundo. We can also search by, for example, if I do maple, you're going to get all the maples show up. This is connected to Vascan, so you can search in French or in English and as well in Latin. So let's say we go back to Acer Negundo, we click on it, and then we just do a search. So when you hit the search button, what happens is it connects through the API to the Postgres database, and then it extracts all the spectral information available for that species, for all the plants for that species. And then it provides you this interface that shows you the name of the species in Latin, the number of plants found, in this case it's 16. And then it creates on the fly this mean spectral signature. So this means spectra for Acer Negundo. So this spectra doesn't exist on the server. It's calculated on the fly as soon as you click on the search button. So you can interact, you can do reflectance. You can click to add the transmittance to it, and then you're going to have the transmittance axis on the right. You see, I don't know if you see well here, you can zoom in and out if you want with the scroll wheel or with If you're on the tablet, you can use your fingers to zoom in. And you see there's a minimum and maximum values for all the 16 plants shown. So there's a measure of variation as well. You can reset the zoom. If you want, you can download the CSV file to reproduce the same graph on your computer with other software if you want by clicking here. So this is for in single species, but it's sometimes not super exciting to look at spectra just for single species because they all look the same. So maybe what we're going to do is we're going to add another species of maple. We're going to add sugar maple, for example. Okay, we're going to add sugar maple here, do a search. And you're going to see for sugar maple, there's 86 plants, and it gets added to this list of species here. Once you have this, you're going to see the two spectra being shown. You can activate one or deactivate it by clicking on the name of the species. You can zoom in and out again if you want to look at the particular area of the spectrum. You can zoom in and out. You can reset it. And that's going to give you a possibility to just keep adding species if you want. As long as you don't deselect them, they're just going to get added. So let's say we clear all of this by clicking the X. The third way of searching by species is just to put something like a genus. So let's say you put the genus, you do a search. This is going to tell you there's 312 species of maples that are found in the data set. It's going to give you the top 5 species. So you see that red maple has 98 plants, sugar maple has 86, and so on and so forth. It shows you the spectra for the top 5 species. There's more than 5, but there is, it gives you a warning telling you that you're only seeing the first 5. Again, we can deactivate some of them just to compare the ones we want, and then we could just keep on comparing spectra like that. The other way to interact, to do the interact in the search would be to say, okay, I want for all the data, you can click to filter by date. You can say, I want all the data for 2018. So you can put the dates for 2018. And you can say, for example, you can combine the filter of date and you can combine it with the search for species. So you can say, I want all the sugar maples for 2018 in the database. And it's going to give you all the maples for 2018. You can also search by location. So that's going to pop you, give you a map. You can click here, and then let's say I'm just want to do the West Island here. You can click OK. You can also draw a more complicated polygon if you want. Once you've done that, you don't have to put in anything for the species. It says search for all species, and then that's going to give us 157 plants. So 157 plants, and it's going to give us the top 5 species found in this area. So we have this showing up here. We see there's one with a particularly odd upper boundary here. I don't know if you see this, but anyway, so there's some of the data. This is using all the raw data from CABO. So we found some of the data that still needed to be cleaned a little bit. So the next thing that you see, the second part of the interface is the map of observations. So if you click on the map of observations, this showed us all in a map form, where all the plants are located. So let's say we click on Cap Saint-Jacques here, and you click on one of those plants. You see that it tells you the information. Obviously, I clicked on one that didn't have a photo. I don't know if they're all not going to have photos. Yeah, so this one has a photo of the plant being, and then you have the name of the site and so on. And if you want to download that spectra, you can click on spectra, you can download the spectra. I'm soon going to allow you to see the spectra for that plant in this window if you want. But right now you can download the spectra. This is going to give you a CSV file with the data for that particular plant only. When you download the spectra, it gives you the raw data for the 6 plants within, if there's like 6 leaves for each plant, it's going to give you the data for each leaf within each plant. So this is really the raw data that you're downloading. So you can explore, and like I said, if there's multiple photos, I don't know, you're going to see a slideshow of the photos. Sometimes there's some zoom, there's some data for which there's, let's go to the beach here. You might have multiple photos for some of the plants. Now you only see one, but you can just explore. Sometimes there's multiple photos and they're just going to be shown as a slideshow. So the third part of the interface is to this is mostly right now for downloading spectra, but so this is all the results. So these would be the 157 plants that are found within the window that I searched for. And then you can just choose what you want. Let's say you just want to download the beach here. You can click on the rows that are just beach. And then you can say download selected spectra. This is I can do it now. I hope this is gonna work because I fixed this only on Friday. Should take a few seconds. And then this gives you a file, a CSV file with the spectra for those 6 plants. And then I won't spend too much time on it here, but you're gonna see that it gives you the 6 plants and then the 6 leaves for each plant. And then it gives you the date, the reflectance, the transmittance, and the raw, the calculated value. So you can technically download if you do clear selection, you can select all, and then you can download the selected spectra. That's going to give you 157 times thousands of rows. Some of these files can get really big, really fast. So I have to find a way, like if you want to download hundreds and hundreds of spectra, this is going to be several hundred megabytes, and downloading them live like this on a web interface is not the best way. But right now it's working. You can also just click on this button here. So you can click on the photos if you want for that species, for that plant. And then you can click on that button to download this particular plant. That should be fast because it's just one plant. Now, the last thing that I just added, I don't think even Etienne has seen this, but I just added now the ability to download by project. So if you click project, for example, Boucherville, we're going to do a search. This is going to give us 73 plants in Boucherville. And again, it gives you the top 5 species and everything for that project, all the spectra. It gives you a map of the field where all the data was collected, and it gives you the ability to download all the information. So this is where I'm at right now with the development of the portal. This is mostly right now for visualizing and downloading spectra. But in terms of next steps, I'm planning to add trait and leaf chemistry data that's also available through Fulcrum, and it's already in the database. So I'm going to be able to add this in the coming weeks. We're going to add the ability to live update data from Fulcrum. As soon as data is entered and is shareable, we're going to be able to see it in the portal right away. Do a bit of performance optimization, several improvements, and then I'm happy to have suggestions because I've been developing this. Obviously, I'm a bit outside of the project. I'm not the user of the project, so I'd like to know how you see what potential you see in terms of improvements and new features and so on. So thank you.